Very consistent at doing the prairie animal and plants of the day because I forget to put it into the daily PowerPoints and then I get in here and I lose my mind. So we're going to try and do better at that for the rest of the case study. So remember, we're doing a grass and a forb and a animal. And so as a result of this, there are going to be more prairie plants and animals in the PowerPoint than we actually go through in class. The only ones we'll hold you responsible for are the ones that we actually talk about. I haven't seen the screencast, but did you talk about C3 and C4 photosynthesis in the screencast?
That is what we're focusing on today.
Okay, so today we're going to talk about C3 versus C4 photosynthetic pathways. So prairie June grass, Kilaria micrantha, is a C3 grass, which means that it has a form of photosynthesis that is less water conservative than C4 grasses do. These are clump forming grasses or bunch grasses. Their grasses all come up from a common root that is very concentrated as opposed to a more rhizomatous sort of grass that spreads through rhizomes. Flowering late in the summer. For a C3 grass, it is fairly drought tolerant, primarily because it has really, really deep roots. You can go online and find pictures of prairie root systems of prairie plants, and some prairie plants have amazingly deep roots. Prairie forb, I have this one growing in my yard. Blue sage. Most forbs are, of course, C3 grass or C3 plants as well. I don't really know a lot to say about it, except that I was about to say it does smell really good, and I know that just from having it growing in my yard. Once again, flowering like a lot of prairie plants do late in the year. So if you come to my yard in the late summer or early fall, you'll see a lot of flowers in my yard still growing because I have a lot of prairie plants growing in my yard. It's pollinated by insect pollinators, bees and butterflies. Very aromatic, and once again, these things have amazingly deep root systems, and this is how they cope with the dry conditions out at Kanza. So Salvia pitcheri. Salvia is the genus of all of the sages for the most part. This is blue sage. The prairie animal of the day is the western meadowlark, Sternella neglecta. There's also an eastern meadowlark that looks very similar, but has a different call and occurs in the eastern half of the United States. This is one of the birds that I think of as a distinctive species on a prairie. You can, when you see them and hear them in particular, and you said you had pulled up the call.
There are 3 different ones.
Yeah, it has a very distinctive call that is.
That one goes on for a while. It's quietness.
Okay, well, let's not do quiet. I don't do quiet. If you hear this on a prairie, for me, that's how I know that I'm in a prairie. Super common bird. If you're driving down I-70 through the Flint Hills, you'll see scats of these things around. They have the bright yellow breast with the dark collar at the top. Okay, stop. Why can I not stop it? I'm navigating around somebody else's computer.
Sorry.
No, it's fine. I just need to be more adept at shifting. So anyway, this is a trench. It has a really broad range in the west. They are feeding on lots of different kinds of insects. They will also feed somewhat on seeds, but mainly on insects. It's a ground nesting bird, which once again is pretty common in prairies because there aren't trees to build nests in. And it happens to be the state bird of Kansas, which is very fitting because it's an emblematic bird. Any questions about these guys? All right, we'll get to other things later. And I'll let you operate your computer because you know your way around it.
All right, so I posted the screencast. I heard from one person that it was clunky. They had a hard time getting it to go. Anybody else have problems actually viewing it? Good, okay.
There was a message on GroupMe that said that you needed permission or something to get access to it. Who was that? Sent that
Yeah,
message?
mine told me I needed permission, and then I just like closed everything out, and then it worked fine.
Okay.
So I don't know if it was just a glitch or like of the app, probably.
Probably, yeah. Okay, so in one of the goals that we had, so these goals from last time one of the goals was to describe where most plant biomass comes from. We never explicitly really told you the answer to that. So, given what you learned about photosynthesis, what reactants go in and what things come out, where do you think plant biomass comes from? Where is all that extra mass when you compare the seedling to the shrub? First?
The surface area of the leaves.
The surface area of the leaves. That is certainly where some mass is deposited, but where does the mass come from to be deposited? So there's conservation of mass, right? You can't make something happen. So where does all that extra mass come from when you're comparing the seedling to the shrub? Asa? Pardon? Light energy? Okay. Certainly light energy facilitates it, but does light energy have mass? Some. Energy does have some mass, but certainly probably not as much as the shrub weighs. That was a good guess. What's being incorporated into the shrub to make shrub? What is a shrub made of? What's a shrub made of? How about the chaparrals? What's a shrub made of? Wait, chaparrals are over there. Sorry. I know, Isaac's got to stand up. Sorry. Elizabeth, what is a shrub made of? Shrub does have water in it, but when we're talking about biomass, we talk about dry weight.
So like it's leaves and sticks and stuff?
Yeah, what are the leaves and sticks and stuff made of? Carbon. Okay, carbon. We're carbon-based life forms on this planet, right? So where is all this carbon coming from?
The air.
The air, absolutely. CO2, right? So when we talk about getting food, right? We're heterotrophs, we eat our food. We're eating carbon-based organisms. And we're taking that carbon, we're taking out the energy, and we're using that carbon as well to build other parts of our cells. Plants do the same thing, except they don't eat, right? So they have to get that carbon from somewhere. They do use light to help them get the carbon. What do we call it when the plants make their own carbon-based food molecules? It was like photosynthesis, right? Yes. Now give me an additional word.
Carbon fixation.
Carbon fixation, absolutely. So the incorporation of that carbon into a molecule with multiple bonds where energy is being stored is called carbon fixation. Basically, we can't use carbon dioxide directly to build building blocks of our cell or to harvest energy, and so we're changing it into a form where we can use it for energy. Carbon bonds are great for storing and harvesting energy. All right, so we've got photosynthesis. You guys have already said two of the things that go into photosynthesis. You said carbon dioxide and light. What else goes in? Okay, water, awesome. Okay, and what comes out? Claire already said glucose and oxygen. Awesome. Okay. And then I had you kind of look at the relationship between photosynthesis and light availability, photosynthesis and intracellular? I think I meant to put intercellular CO2 concentrations, so CO2 within the leaf. Okay, so here's just a reminder. Autotrophs have to make their own food, so we have these 3 stages. We've got photosynthesis, where you use this light energy together with these smaller molecules. Where are the smaller molecules coming from? What are these smaller molecules in photosynthesis?
That'd be CO2, water.
Yeah, CO2. We're going to have some RUBP, right? Okay. We're going to take energy and smaller molecules, and we're going to we're going to make food. We just said that food is glucose, right? Okay, so that's photosynthesis. Then we're going to do the opposite of photosynthesis, and we're going to break that food down to get energy. Why do plants not just use the energy directly? Why do they go through the process of making the large carbon molecule, breaking it down, and then using the energy again? Why not? These 2 steps look the same, yes? Why do this and then that and then that? Why not just do this? Why go through all the effort? Transporting it through the plant. Tell me more about that.
Because you can't really transport the energy without carbon monoxide.
You need something to carry the energy. Absolutely. So we need an energy carrier. You're going to collect it. If you're going to collect a bunch of stuff, you can't just use your arms. You need a bucket, right? We need an energy carrier. And when you get to cell and molecular, you'll actually view how if you are using energy directly and you harvest it all at once through this kind of process, that's what fire is, right? If we didn't harvest our energy just a little bit at a time via different energy carriers, we would literally burst into flames. We would burn up. So we need to store it so that then we can harvest it just a little bit at a time. All right, so today we're going to talk about C3 and C4 photosynthesis. We're going to, our goal is to be able to describe, compare, and contrast those 2 types of photosynthesis. And then, based upon whether a species is C3 or C4, be able to predict which one will have higher photosynthesis in particular environments. And we'd like you to be able to list and explain the primary differences between species that use C3 or C4 and how they interact with global change. Now, I don't know how far we'll get on global change today, but hopefully we'll be able to at least start it. All right, so what we talked about in the screencast, that's actually C3 photosynthesis. That was the first kind of photosynthesis that was discovered because it's the most common. C3 photosynthesis. So what's that little weird kidney bean shape that sort of houses all of that? What is that representing? It's not a cell, right? It's not the mitochondria because the mitochondria does the opposite of photosynthesis. It breaks down. It's chloroplast. Excellent. Good guess, though. Mitochondria often are shown as these bean-shaped things, too. Okay, so this is the chloroplast. What kind of cell is the chloroplast in?
Plant cell.
Plant cell. What kind of plant cell? No. The chloroplast itself probably originated from a cell, but is no longer a cell itself.
Is it the mesophyll cell?
Yes, the mesophyll cells. So inside the mesophyll cells, there's a bunch of photosynthesis going on. Inside the mesophyll cell is a bunch of chloroplasts. And inside the chloroplast, we've got stroma, and we've got these stacks of thylakoids. Okay? So we've got light reactions. So light comes in, and those light reactions are happening on and across the membranes of the thylakoids. The light reactions split water, we get out oxygen. We're making some ATP and NADPH from NADP and ADP. What would you call these molecules in here? Bryce, you know you want to say it. Energy carriers, absolutely, absolutely. And specifically, this guy is basically carrying an electron. This guy's carrying an extra whole phosphate group. I think I mainly was calling them electron carriers, but so they're high energy carriers, right? These guys can carry energy, and that's what they're doing, is they're taking this energy that's passed through here, they're catching that pass, and they're taking it over to the Calvin cycle. Have we made any glucose yet? No. Okay, so we need to actually fix that carbon. We don't have a carbon source yet until we get over to the Calvin cycle. That's where CO2 comes in. Okay, so CO2 comes in. We're going to use some of this ATP and NADPH, and we're going to use an enzyme called Rubisco to add CO2 to our UPP to make a 6 carbon sugar as it comes out. Okay, C6H12O6. All right, this is C3. We're going to talk a little bit about C4 today, but before we get to that, I want to go back a little bit and talk about this relationship between photosynthetic rate and light. All right. So photosynthetic rate, you can think of as the rate of carbon fixation that's happening, right? And how would you describe this relationship? How would you describe it?
Of the photosynthesis and
Between the amount of light that's coming into the plant versus the amount of carbon that can be fixed.
what it shows, you're gonna have a point where if it's not really productive to have any more. You mean, some plants can handle more than others, so they're gonna be able to take in different amounts of carbon and sunlight, but they can only process so much of it. They only need so much of it.
I don't know about they only need so much of it, but they can only process so much of it. Okay, so you're gonna have a direct relationship. The more light you get, the more carbon you can fix, given an infinite supply of carbon, until you get to a certain point. And there's a certain point that plants reach where more light becomes actually damaging to its tissues, right? And so it could continue to fix carbon if its tissues weren't being damaged. Okay, so at some point, the light gets so intense, right, you're cooking the plant. You don't want to cook the plant anymore, right? Okay, so basically the more light you have, the more photosynthesis you can do until you get to this maximum thing where you're starting to damage the plant. Okay. All right. We talked about how uptake of CO2, I think, is driven by I think we talked about it, not uptake is, I think, driven. Uptake of CO2 is driven by a concentration gradient. So when we're talking about a concentration gradient, what do we mean? Kennedy?
So essentially, it's typically talking about like the inside of the cell versus like the outside, and then there's a higher concentration or lower concentration, and you move like the concentrations around basically to like equal them out, but it has to have some type of higher and lower in order for it to move.
Great, absolutely. So when we're talking about concentration gradients in living systems, typically there's some kind of membrane, and then your solute, whatever you're interested in, if there is a gradient that exists, a differential in concentration, then it's going to be more concentrated on one side than the other. Now from chemistry, and diffusion, what do you know about starting out with a permeable membrane and having a high concentration of stuff on one side and a low concentration of stuff on the other side? What's going to happen? Chris?
It will eventually become equal.
It'll eventually equalize, right? And does it need the input of energy from a system? Does it need something working to make that happen? No, you don't really need a little guy in here shoveling these particles to the other side. Actually, the lowest Gibbs free energy is for both of them to equalize across that membrane. Okay, well the same thing is true of CO2 and leaves, okay? So we actually have pretty low CO2 in the atmosphere. It's less than 0.04%. But inside the leaves here, it's even lower, typically, if photosynthesis is happening. Why is it even lower inside the leaf if photosynthesis is happening? Isaac?
Because all the CO2 is being used up for photosynthesis.
It's being fixed, right? So it's being pulled out of this extracellular space into the mesophils, and being fixed in the chloroplasts, right? And so as long as it's being taken out of this space, it's going to go in through here. All right. And the more CO2 you have, the more carbon fixation you can do because you have more carbon, right? Until you run out of other reactants, like water, Rubisco enzyme is maximized, that kind of thing. So why don't plants always open up their stomata, always, as much as possible? Why do we even have stomata? Why don't we just have big holes? And to remind you what I mean by stomata, it's these guys here, right? And they can open and close. Why don't we just leave them hugely open all of the time? Bryce? Teeth from losing water. Okay, well, why do we care about water? Why is that a bad thing? I mean, part of the answer to that question is kind of like duh, but the other part is not like duh. So tell me about why losing water would be a bad thing for a plant.
You need water for the light reactions in photosynthesis.
You need water for the light reactions in photosynthesis, so it provides that those extra hydrogen, those protons, those extra hydrogen atoms to build up the proton gradient in the electron transport chain. Good. It's also important for a lot of other physiological things, right? You ever seen a plant that hasn't been watered in a while? What does it look like? Wilted. If it's been watered, not been watered in a really long time, it might even be shriveled or brown. The first thing they do is wilt, right? Why are they wilting? It's not just that they don't feel good. What's the physics of water at that point?
So we're losing the pressure within the cells that keeps the cells expanding and keeps the energy.
Yeah, so plants have their shape because they have turgor pressure. They have water pressure inside their cells that keeps them like inflated. It's kind of like if you were inflating a pool toy, except that it's with water, right? Inside this leaf here, this is almost 100% humidity. And that's what it needs to be really functional, right? And so if you lose so much water, you're gonna start deflating as a plant and starting to be really loopy. And obviously that's really bad for a lot of physiological reasons as well. One, the plant can't cool itself, two, other molecular reactions can't happen. That would be bad. So we don't want to lose water. So that we need this balance between losing water and taking in CO2. All right. So CO2 is following these laws of diffusion. If the water vapor is 100% humidity in here, under most conditions on Earth, where is it going to diffuse? Out, right? It's going to go out because the air is usually less than 100% humidity. Okay. And then the CO2, as long as photosynthesis is happening and carbon keeps getting taken up and fixed by the cells, then the CO2 concentration in here is going to be less than outside, and so it will keep sort of sipping that CO2 up. So we need to maximize our water use efficiency, the ratio between gaining carbon and photosynthesis and losing water. The loss of water through stomata is called transpiration. You guys have heard of evaporation. This is transpiration. This event is transpiring. That's a different use of the word transpire. Water coming out of the stomata is transpiration. So we need to minimize transpiration while maximizing carbon fixation. All right. I accidentally left that title on there. I'm not actually going to ask you what limits photosynthesis right now. But I do want to point out this enzyme. I'm not going to make you learn all of the enzymes in the light cycle and the Calvin cycle, but one that you really should know is Rubisco. This is ribulose bisphosphate carboxylase oxidase, I think. Rubisco. Rubisco is the enzyme that will actually grab CO2 and react it with RUBP to make a 6-carbon molecule. RUBP is 5 carbons and it hangs out over here in the cycle. It's going to be added to CO2 by Rubisco. So Rubisco is doing this job. But Rubisco is not very faithful to CO2. Rubisco is kind of getting bored in the relationship sometimes and likes to step out. Sometimes Rubisco is like, hey, oxygen is looking pretty fine right now. And if there is a greater amount of oxygen than CO2 in the cell, or if there's too high an amount in the leaf, it will actually bind with oxygen. And that's a bad thing because carbon dioxide does not get fixed. In addition, this is kind of small, but here we've got our RUBP. Okay, this is that molecule that, if under normal circumstances, it's going to be added to CO2 by Rubisco, and it's going to become this 3-PGA, which will eventually make sugars. But in the case where there's too much oxygen around, Rubisco will actually add oxygen to RUBP, and instead we get glycolate, which not only doesn't make glucose, but it also steals RUBP. It doesn't allow it to go back into the pathway. So when we have high temperatures or really high oxygen concentrations, Rubisco will actually favor this process of binding CO2 with RUBP called photorespiration. It'll favor photorespiration over photosynthesis. Kind of doing the opposite. It's even taking the carbon molecules that exist in the system before the CO2, and it's using them in something that is not useful to the plant. Now, photorespiration is not always a bad thing. Photorespiration can prevent damage when there's too much light. When there's too much light coming in and there's not enough enzyme available, or when the heat gets too much because of that light, it can this process of photorespiration can occupy excess oxygen and cool down the plant somewhat. But it doesn't produce any ATP, doesn't produce glucose, and it results in a loss of CO2, loss of carbon. It can't be fixed. Where would you expect to get really high temperature or oxygen concentrations? Like if you think of a community or an ecosystem, what kind of community would you expect to find those? Elizabeth Quinn, where am I going to find high temperatures, a plant having to deal with high temperatures? The desert, absolutely. Okay, the desert. What defines a desert?
Precipitation.
Low precipitation. Okay, what else defines a desert?
Drastic, temperature changes, so you have high heat.
Right, so you might get really variable temperature changes. When you think of desert, you probably think cactuses and sand and stuff, right? Okay, so that's the classic desert. But we can have areas that are really dry that aren't like that too, right? In fact, the prairie is really dry seasonally. And so you get this drought stress. And in this case, this C3 photosynthesis is not so great. Because when you're having drought stress or high temperatures, you're going to photorespire. And that's going to mean that you can't fix carbon at your maximum rate. I want you to keep in mind that nitrogen is pretty important to plants. But up to 1/3 of the nitrogen in plants is in this one enzyme, rubisco. And so that's a lot of why nitrogen is important to plants, is because you need rubisco to fix carbon, yeah? Okay, keep that in mind for later when we're thinking about Kanza prairie. All right, so now let's put on our thinking caps. We're going to be engineers real quick. And if you were going to design a plant such that you would prevent photorespiration and water loss, or reduce it, what could you do? Starting from a C3 photosynthesis plant, how could you change that plant and that cell and how it works to prevent water loss and prevent photorespiration? You can think on your own, but also think together with your groups. Take a few minutes, talk about, if you're an engineer, you're going to re-engineer C3 plants, how could you physically or chemically overcome this limitation by changing the plant somehow? Now remember, you're going to want to overcome water loss and photorespiration, but you also want to still keep fixing carbon too, right? Okay, I want one idea from each table, and that means the faster you put your idea up, easier it's gonna be. Right? Because by the time 3 tables put their ideas up there, there's not gonna be much for you to choose from. The number of ideas in the universe that are not seen. All right, looks like we've got at least one idea from each group. We have keep stomata openings closed. Good. That will prevent water loss. What will that do to your CO2 uptake though? Down. Okay, so yes, this is smart. So if you are a plant and the sun comes up and it starts getting really hot, you can close your stomata during that hottest part of the day. Okay, so that means that all else being equal, during that hottest part of the day, you're not getting a ton of carbon fixation going on. What's happening though with light availability during the hottest part of the day? It's high, right? And so you're kind of missing an opportunity there, right? If you've got high light availability, we know that photosynthetic rate goes up with light availability. So we want to hop on that.
But aren't light reactions and the Calvin cycle independent of each other? So one could do the light reactions during the day and then the Calvin cycle at night?
Tell me what you mean by independent of one another.
Well, the Calvin cycle requires it has for its reactants the products of the light reactions.
Sure.
So during the day, the plants use all that light that they're, or water that they're not releasing back into the atmosphere because of the water and all that light that's coming down on them to get that ATP and the NADPH, and then at night they'd open the stomata so that they can get CO2 in and fix it during the night when they're not losing as much water.
Okay, so what I'm hearing is a separate way to get around this, which is to actually separate temporally, in time, separate the light reactions from the Calvin cycle. Okay, so do them at separate times. All right. Okay, so we could then use that water during the day. Sure, we could use that water during the day, keeping our stomata closed, and then open them at night to get CO2 and do our Calvin reactions. Cool. Okay, so I'm gonna add this. Keep the stomata openings closed during the day. Okay, that's a good plan. Okay. So another option we have is the ability to store or turn off Rubisco. Tell me a little bit more about that, guys.
So, like, in the Calvin cycle, like, if the Rubisco binds to oxygen, if there was a way for the plant, like, if they didn't have enough carbon, if they were able to just, like, store Rubisco, so it could be used when they had available carbon, or, like, turn it off when there was too much oxygen, so it wouldn't use the oxygen instead of the carbon.
Yeah, okay, so maybe modify Rubisco itself, or come up with something else that would maybe bind Rubisco or sequester it in response to when the ratio of oxygen to carbon dioxide is too high. Okay, and so this would be some sort of bridge. What if you could, let's continue with the storing aspect. What is the problem with Rubisco? What causes the photorespiration? Asa?
That's reacting with oxygen.
It's reacting with oxygen, right? So if you could somehow isolate it from oxygen, maybe, physically, then that might be another way that we could store it away, right? Okay. This is a good one. This is a good, like, genetic engineering thing. Alter Rubisco's preference to oxygen. Make it where it doesn't really like oxygen anymore. It's a great idea.
Then it would just be Rubisco.
It would be Rubisco. It would be, it sounds like some sort of crispy treat. Sorry. Water storage. Let's talk about water storage. Is that you guys back there? Or who was that? Water storage. Who suggested? That was you guys. All right, tell me more about water storage.
We were saying, like, deeper roots on the plants to prevent water loss.
Okay, deeper roots would allow it to accumulate more water, certainly. What about, how's the evapotranspiration gonna change if I'm a super thin leaf versus a giant barrel leaf? Okay, so think about the surface of the leaf and how much water it's going to lose through stomata if it's a super thin, flat leaf versus a really fat barrel. The super thin, flat leaf has a lot more surface area compared to its volume, right? And then the big fat barrel has more volume to its surface area.
So that's why succulent plants have those huge leaves.
Where do we see huge barrel-shaped plants? In the desert, right? You see cactus, right? And so the type of water storage that can help you to overcome water loss is to limit your surface area. Okay, but also in the desert, there are other things playing in. You've seen trees in the desert? They do exist, trust me. They have little tiny flat leaves, all over the place, right? So there's a balance between using water and also then just being able to photosynthesize. Waxy cuticles is also a really great one because you're going to collect a little bit of light and you're also going to reduce transpiration, right? But then again, you get sort of this balance, this trade-off. Okay, these are great ideas. And in fact, C4 plants decided, you know what? Evolution and ecology class is really smart. I'm gonna take their advice. And you know what? I am going to do some sort of weird combination of these things. I am going to, not only will my plants maybe develop ways to store water or have a waxy cuticle so that we don't lose it as much, I'm going to isolate Rubisco, and I'm going to use something that doesn't have such a great affinity for oxygen. Okay. In C4 photosynthesis, I want to orient you to what you're looking at real quick before we talk about how the photosynthesis is different. Here we actually have two separate cells, and what they've ignored is that they haven't shown you that these are not just two separate cells. This is what you're looking at here would be happening in the stroma of our chloroplast in the mesophyll cell. And these would be happening in a bundle sheath cell, okay? So we have spatial separation in C4 plants. We're gonna capture our CO2 in one spot, and then we're gonna fix the carbon in a different spot. So if you capture the CO2 in one spot, and we can have oxygen in that area, it doesn't really matter. Then we're gonna move it to a different spot where we can do the fixation in a place where oxygen is not at a high concentration, okay? These are two separate cells. They have these little tunnels between them. This is a cell wall, okay? And they have these little tunnels that indicate transport passages. Are the light reactions going to be really impacted by this? Are they going to be the same? Are they going to be different, you think? What would you predict? Kelsey, what do you think? Think the light reactions will be impacted by this or not? No. Light reactions are the same. They're still there, but they're not shown in this particular drawing. We still need ATP and NADPH for carbon fixation. And that light reaction is still occurring, but it's not shown in this figure. I want to just point out, though, that it is still there. It's still happening. Where is the carbon dioxide diffusing in, or is it diffusing in? Is it happening in this bundle sheath cell? Is it happening in the mesophyll cell? It's a harder one. It's initially being taken up by the mesophyll cell. Just the way it would in C3 plants, it is diffusing in. What's my next slide? Is it initially being captured by Rubisco, though?
Yes.
Let's see, I've got CO2 and Rubisco over here. So it is eventually.
Not initially, otherwise we have photo respiration again.
Not initially, or we'd have the problems of photorespiration. So initially it's coming in here, and it's being captured by an enzyme called PEP carboxylase. Carboxylase is this carboxylation, meaning adding carboxyl group, right? So COO plus an H. This is carbon fixation is often called carboxylation. That's what Rubisco does as well. PEP carboxylase, though, doesn't care about oxygen. It's married to CO2, it's happy, doesn't have to step out. It's doing good. Okay. PEP carboxylase is not as likely to bind oxygen. Now, I want to just compare and contrast. Here's regular Calvin cycle, C3 photosynthesis. Here's the first part of what we were just looking at in C4 photosynthesis. We have CO2 coming in. Rubisco, this enzyme, is adding that C to a 5-carbon molecule, RUBP, to make a 6-carbon molecule. That's the fixation right there. fixing it from gas to this 6 carbon. A separate enzyme cuts that in half to make 3-PGA. Okay, this carbon fixation is right here. In this case, CO2 comes in, and carboxylase fixes it. It takes PEP, it adds the CO2 to PEP, and you get oxaloacetate right there. And so this is only a 4-carbon critter. Glucose is a 6-carbon molecule. Are you closer to making a 6-carbon molecule from a 6-carbon molecule or from a 4-carbon molecule? A 6-carbon molecule, right? And so this actually isn't quite as efficient in photosynthesis. You can get away with not photorespiring, okay? So you're still able to fix carbon, you're not wasting all the CO2, but it's not quite as efficient, okay? All right. So I'm not gonna make you learn all of the names, but you should know Rubisco, you should know PEP carboxylase, okay? Note that in this, we've got ATP going in, NADPH going in, same as here, okay? Still using those products from the light reaction. Okay. And then you just, fixed CO2 then goes into the bundle sheath cell, and a little bit more CO2 is actually, the CO2 is liberated from malate, which is made from this oxaloacetate. So you fix the CO2, you liberate it, you fix it again. But you've got that spatial separation so that oxygen is not in there, and you've got a separate enzyme that is less inclined to bind with oxygen. All right, I want us to stop for a second. I want you to turn to one other person. If you've got an odd number at your table, turn to a person at a different table. And I want you to take turns explaining the differences between C3 and C4 photosynthesis. Do that for just a couple of minutes.
C3, CO2 goes directly to the crystal. And C4, have carbon number takes it, includes it,
Explain the difference between C3 and C4. So walk through C3 and then walk through how C4 is different.
Is this part okay, so the only difference there is that it's just in a different cell. So that
and remember the O, so it's ribulose phosphate
So the nice thing about it is you have long-term everything's happening.
C3 versus glycolate. Yeah.
So all the difference comes from this place. What's that all about? It's just in the bundle she sells.
Sorry.
The bundle she sells are protected from the effects of oxygen. So you don't have competition of oxygen in carbon or access to Rubisco. You see here that Rubisco is still in there.
Was not successful.
It's just that no oxygen, just carbon. Carbon and oxygen are both here, but what's the enzyme that you use to capture carbon here? Carboxylase. It's important to know the names of things sometimes. It's not always important, but this is one of the times it really is.
Yeah. Yep. Yeah, and CO2 is like super packed into that bundle sheet cell.
You notice that she spells it as a thing?
But also, there's lots of water needed in the light reactions as well too. But certainly you have to have this nice big storage compartment in these bundle sheet cells. Okay, I'm hearing the noise quiet down a bit. Please continue to ask me when you have questions. The next thing though that I'd like you to do is to open up iTunes U. And there's a worksheet on C3 and C4 plants. Before you do that, Dr. Kowinski, were you gonna?
Yeah, it's just one of those situations where not always do you need to know what an acronym stands for, but in this case, it actually helps you.
Pretty helpful.
Because ribulose bisphosphate, that's the 5-carbon compound that in carbon fixation you add carbon dioxide to make it an unstable 6-carbon compound that then breaks down to 2 3-carbon compounds. So this basically tells you the starting point.
Mm-hmm.
So to ribulose bisphosphate, you can either carboxylate it, or you can add oxygen to it.
Oxygenate it.
PEP carboxylase, this once again tells you Pep C. We sometimes call it Pep C because that's just easier. The starting point here is phosphoenol pyruvate, which is a 3-carbon compound, and all you can do to it is carboxylate it. You can just add a carbon dioxide to it. And so if this was a problem, if Pep C had a problem with oxygen, it wouldn't be called Pep C, it would be called Pepco.
Right.
But it's not called Pepco because it doesn't do the thing with oxygen.
Right, and so when you're trying to memorize these and keep them straight in your mind, you're going to think, okay, both of these enzymes can fix carbon, but one of them is not able to oxygenate. Okay, so Rubisco is a carboxylase or oxygenase. That's why it's called carboxylase oxygenase at the end.
So Pep C can be hanging out in the mesophyll cells. And it doesn't matter how much oxygen is out there because it's just not going to play around with oxygen.
Nope.
It's just not that.
He's not that dude.
Or
Or
that
lady.
dude. It just doesn't get around like that.
No. Oxygen likes to get around. Pep C's not having it. You will learn through cell molecular oxygen really likes to get around. Okay, so what I want you to do is I want you to open up that worksheet and I want you to look at it a little bit, just for a second. I want us to look at it together.
I didn't know that you wrote Rubisco the same way I write Rubisco.
Yeah.
I I've written Rubisco that way since undergraduate because for me
It's super
important to-
handy, actually.
To understand what it is doing.
Yeah, it's more than just nomenclature. I mean, it is nomenclature, but it's just super handy to write it that way because it just fits in your brain better. All right, so in this worksheet, we're going to think about a C4 species, Andropogon gerardii, this big blue stem, and Salvia pitcherii, okay, this blue sage. Now you guys you've both seen those, right? So we saw blue sage today, and we saw this big blue stem on the very first day that we started talking about prairie critters, okay? Both of the species occur in the tall grass prairie at Kanza, and Kanza is characterized by high light availability, and seasonally, it has water stress. What season do you suppose it has water stress in? Summer's pretty dry, right? Particularly late summer, right? Okay, and so you're going to want to go through and think about how these things are C3 or C4 plants. You're going to want to ask, okay, is high light availability going to favor one or the other? Is low water availability going to favor one or the other? And based on those responses, not based on those responses, based on this table may help you with those responses. So this table is asking you about, for a C4 plant, which enzyme catalyzes the initial, that first capture of CO2? Where is the Calvin cycle or carbon fixation located? What is the possibility of photorespiration? Is it high? Is it low? So we still have some time in class. I want you first to fill out this table, and then I want you to answer those questions above it. Work on that right now.
Discussing the method section of the skill for guys and have your drafts.
For whatever reason, I spaced it.
I knew it was on the schedule and I see.
They do have it to turn it in for sure.
But no sense in turning maybe it was on Friday. You have a question?
You may do this in your notes, or separate. I mean, I would prefer that you do it in your notes. Yeah.
So this table is answering the questions you guys have.
Right, so do the table first, and that'll help you with the questions above it. And, you know, I looked my second email just and when you think you've got some good answers to those questions up above the table, turn to your neighbor and talk about what they think. Oh, haha, never mind, we have 1 minute. Okay, please finish this for next time. We will open class by discussing this. It's basically prep homework for next time, okay? But here's here's what else, okay? You had a lot of things due today. On Monday, I was unable